So I'm going to start in a way that I often used to. I haven't for a while, but I think it's an important overview, especially for a group that's being introduced to the subject. To start, and do you see my pointer, by the way? Yes? Yeah, okay. So to start with some very basic concepts that were widely discussed in the early 20th century that still in some ways root our thinking about atrial fibrillation. So there are essentially three opposing schools. Engelmann and Winterberg were the proponents of the notion that AF was maintained by one or more rapidly discharging focal ectopic beats or sets of beats, local ectopic foci firing away quickly. Whereas Lewis thought that AF was due to a single organized reentry circuit, and Gary and Mines were proponents of a notion of irregular, virtually random reentry coursing through the atria and finding non-refractory, finding excitable tissue at all points in their paths such that the arrhythmia was self-maintaining. For the ectopic focus and single circuit reentry notions, you have to have an additional idea to explain why the surface electrocardiogram shows completely irregular atrial activity, or apparently completely irregular. And the idea to explain that was simply that these, either the focus or the single circuit, were discharging so rapidly that the rest of the atria couldn't keep up one-to-one. So that the conduction waves would break up when they hit refractory tissue and then conduct irregularly over the atrium. One of the classical demonstrations of the notion of an ectopic focus maintaining AF is an experiment that Gordon Moe did. He applied a crystal of aconitine, which partially inactivates sodium channels and causes very rapid local firing to a dog's atrium that produced very rapid, clear atrial fibrillation. And when he took the crystal off, or he applied a clamp around the area where the crystal was situated on the atrial appendage, the fibrillation stopped immediately. A simple example you can do in an experimental lab is use a pacemaker to induce atrial fibrillation. And typically, especially in a normal atrium, the fibrillation will last as long as you're rapidly firing the pacemaker at a rate the rest of the atrium can't follow. As soon as you stop, the fibrillation will fairly rapidly disappear. So the subsequent thinking about AF in man has followed the lines of a number of components that determine its occurrence. So the first is AF initiation. The idea is that AF has to be initiated to start. And in fact, studies of patients have shown, patients who are monitored continuously have shown that AF usually begins with a single atrial premature complex that triggers fibrillation. Then you have a process of perpetuation that maintains it. And finally, over time, there's a subsequent process known as progression, in which the nature of the atria change such that AF becomes more and more sustained and harder to stop and keep stopped. So if we think about the initiation of AF, it acts on a vulnerable substrate, typically to induce reentry, which is responsible for the perpetuation. And then this substrate changes with time to produce progression. So I already mentioned the proponents of the role of ectopic foci, and I would point out that these three theories are not necessarily mutually exclusive. you can have the involvement of ectopic foci, which may or may not maintain the AF, but at least act as triggers. You can have single circuit reentry early on. You can have multiple circuit reentry as a final common pathway. So they're not mutually exclusive. So what is the role and mechanism of ectopic foci involved in AF? So the ectopic focus idea became sort of not very popular for quite a period of time, from the period essentially Engelmann and Winterberg until Michel Asaguer's seminal observations published in the New England Journal of Medicine in 1998, in which he described the series of patients in which AF was clearly coming from a focal source in the atrium, and he could apply. So this is a fluoroscopic image. You can see the pulmonary vein and this focal source, the location of the focal source within the vein. In this particular case, it's fairly peripheral in the pulmonary vein. Often they're closer to the body of the atrium, but you have focal sources in the pulmonary vein that produce ectopic beats and that can in themselves maintain AF. So this was a very seminal observation because it exposed a novel anatomical basis of AF that hadn't been suspected before, and in addition, restored the notion that ectopic foci could play a critical role. And critically, it introduced a therapeutic maneuver because Michel and Pierre ablated at these sites and stopped the AF. Now, pulmonary vein directed procedures have evolved since then. They've gone from trying to attack a source to isolating the pulmonary vein. A question for us from a pathophysiological point of view is why the pulmonary veins? We'll come back to that at the end of the second hour of this class. So now we see that the pulmonary veins can be involved. Michel's observations suggested that they can actually be involved in maintaining AF as a continuous, rapidly discharging focus. And they can also be involved as a trigger acting on this vulnerable substrate, which allows for reentry. And this is a schematic diagram of reentry. We'll come back to the principles more later on, which then result in perpetuation of the arrhythmia. And as I mentioned before, we have this notion of progression. So what is the physiological basis of spontaneous activity, ectopic activity in atrial cells? So the key, a bunch of experiments had identified different potential mechanisms in animal models, but I think the key observations here were from Dobromir Dobrev's group, which has worked very extensively with human atrial cells. So they isolate atrial cells from samples taken from the operating room from patients in various clinical categories. They may be normal, they may have heart failure, they may have atrial fibrillation, they may have sinus rhythm. So this first study was published in Circulation 2012, and they isolated atrial cells from patients in sinus rhythm and atrial fibrillation, which I'll show you in a moment. And they recorded intracellular calcium using a ratiometric dye, which allows for precise calibration and measurement of intracellular calcium concentration. At the same time, they recorded transmembrane potential. And so you can see here what you're looking at is a series of fired transmembrane potentials. So the cell is being paced. It's from a single cell. We see its resting potential is about minus 80 millivolts going up to plus 50. It's, well, I'm not, yeah, I guess so. And it's being activated repetitively by the pacemaker, and then the pacing is stopped. These are the corresponding calcium transients, with each firing, calcium enters the cell, and that, of course, is what produces cellular contraction. And at the end of this period of pacing is stopped, and you see basically quiescence. In cells from patients with atrial fibrillation, they noted spontaneous depolarizations, which were not seen under control, corresponding to calcium transients. And they believe that these calcium transients were triggering the action potential, and we'll see in a moment how that could happen. So they went on to analyze the underlying mechanisms. So in this schematic diagram, I'm going to explain a system that's somewhat complex, but it's the real system. And so when calcium is stored in the cell in an organelle called the sarcoplasmic reticulum, indicated here by SR. The job of the sarcoplasmic reticulum is to kind of be the policeman of intracellular calcium. So it puts all the calcium in jail and diastole so that the intracellular calcium concentration is very low and the cell can relax. Then when calcium enters in systole, the policeman lets a bunch of calcium out of jail, in fact, a lot of it, and that substantially increases intracellular calcium concentration, activates the actin-myosin interaction and causes cellular contraction. And then at the end of systole, when the action potential ends, the calcium is taken back up again. Now, one critical part of the machinery for this is the ryanodine receptor. The ryanodine receptor is a membrane ion channel. It's called the ryanodine receptor because it was first purified by its interaction with the toxin ryanodine. That's the only thing we need to know about ryanodine for the moment. So it's the name of this ion channel, and it's called a receptor, unfortunately, which is confusing because it's really an ion channel. It's a calcium sensitive ion channel because the job of the ryanodine receptor is to sense the increase in intracellular calcium that happens when the action potential occurs. Calcium enters when the cell depolarizes, and then it greatly amplifies that signal by allowing calcium out of the SR. It opens up, allows the calcium to go out of the SR. Tell me if I'm going too slow, but I'd rather explain things in more detail. And I don't know, is this elementary for everybody, or is it worth going into this level of detail?
The students should be aware of all of this, I think, because it has been repeated many times, but follow your pace, do as you want.
All right. Well, I'll do it like this, and you'll see we'll get into pretty heavy stuff fairly quickly. Anyway, so the ryanodine receptor then releases calcium and takes it back, it opens up to release calcium in systole. It then closes in diastole, and the calcium is taken back into the SR, where it has to be returned via the sarcoplasmic reticulum calcium ATPase or the SR calcium pump. So what the life cycle of calcium is such that the released calcium has to be gotten rid of. So one way it's gotten rid of is, so this is now in systole, the intracellular free calcium has gone up. It has to be taken back into the SR to restore the SR stores, but also it's pumped out via the sodium calcium exchanger, which is electrogenic. It pushes in 3 sodium ions with a net charge of +3 for each calcium ion, with a net charge of +2 that it pumps out. So each cycle of the exchanger moves one more positive ion in, then out, and it generates a positive intracellular charge, which will depolarize the cell. So what causes spontaneous after depolarizations, which are believed to be the mechanism by which released calcium depolarizes a cell and leads to ectopic firing, is shown as follows, and it's due to a spontaneous excess release of calcium, to a spontaneous release of calcium during diastole, when normally it's not released. So it's due to, in a sense, an aberrant reopening of the ryanodine receptor. Now, that can happen because of one of three things. So the ryanodine receptor, as I mentioned, is sensitive to calcium. Calcium is the signal for it to open in systole. But if the intra-SR calcium gets too high in diastole, it goes above a threshold and causes release, opening of the ryanodine receptor and release of calcium from the SR. So one of the possible causes of atrial ectopy is cellular calcium overload. A second cause is an abnormal SR. So it's normal for the SR to increase in the SR calcium to increase in diastole as the calcium is being taken back up, but the normal level doesn't normally trigger an aberrant release, an aberrant opening of the SR. However, if the an aberrant opening of the ryanodine receptor. If the ryanodine receptor is hypersensitive, even a normal restoration of calcium levels could theoretically trigger a release of calcium from the SR. Or, we have a fluctuation of calcium under different physiological conditions, simply to an exposure to catecholamines, which increases SR calcium, or to fast heart rate, which increases SR calcium, could be enough to cause release of calcium from a hypersensitive ryanodine receptor. So there are two factors that could make the ryanodine receptor hypersensitive. One is if there's a mutation in the receptor, which makes it too sensitive. That's the cause of a syndrome called CPVT, which you may have heard about. It's a congenital ventricular arrhythmia syndrome. The other is if the ryanodine receptor is hyperphosphorylated. During normal physiology, the ryanodine receptor gets phosphorylated both by the direct action of cyclic AMP through protein kinase A, which is a phosphorylating enzyme, or via an increase in calcium through calcium calmodulin kinase type 2, CAMK2. And these phosphorylations of the ryanodine receptor increase its sensitivity and can make it leak. The final factor to consider is the sodium calcium exchanger. If it's abnormally or excessively active, that can cause an excess current in response to a given release of calcium. So what happens in AF patients? So the first possibility that Niels Vogt, who is the first author of this paper, looked at was the possibility that there's ryanodine receptor dysfunction. And to do this, he measured the spontaneous release of calcium by looking at the effect of tetracaine, which blocks calcium transport, and looking at the exchange in current after a set of, looking at the change in calcium after a set train of pulses. So here you see there is under voltage clamp conditions, there's a decrease in intracellular calcium, which reflects the calcium leak exposed by tetracaine. They then triggered the release of most of the calcium from the SR with caffeine to measure the SR calcium load, because the more, as I mentioned, the more calcium there is, the more leak there will be. And so they normalize these. And then looking at comparing sinus rhythm patients with atrial fibrillation patients, they saw that they had an enhanced SR calcium leak, which tells you that the ryanodine receptor is leaky. There's something funky going on with the ryanodine receptor that's causing an excess leak. By the way, this leak was blocked by KN93, which is a blocker of CAMP kinase 2, and not blocked by KN92, which is an inactive analog, also not affected by H89, which is a protein kinase A blocker, pointing to CAMP kinase hyperphosphorylation of the ryanodine receptor as the cause of this leak. And they went on directly to measure the phosphorylated components and saw an increase in the relative phosphorylation at the ryanodine receptor specific site, serine 2808, as well as some increase as well at the less selective 2814 site. So CAM kinase, this points to CAM kinase mediated ryanodine receptor phosphorylation being increased in chronic AF, which is they define as AF more than 6 months, lasting more than 6 months, and that being the cause of the calcium leak and possibly the delayed after depolarizations. So ryanodine receptor dysfunction due to CAM kinase mediated hyperphosphorylation, which causes leakiness of the ryanodine receptor, appears central. By the way, I didn't mention ryanodine receptor is typically abbreviated capital R, lowercase y, capital R. Okay, so what about the sodium calcium exchanger? What happens there? Well, they measured sodium calcium exchange current. There's a way to do that by controlling the intracellular and extracellular contents and causing an action potential while voltage clamping and looking at the change, while calcium controlling and look at the change in current. And they also looked at the slope of the sodium calcium exchanger against the calcium content, which is a way of directly assessing its activity. And you see that sodium calcium exchange activity is increased, as is the protein, as shown by these Western blots. These blots represent the expression of sodium calcium exchanger in AF patients, which is enhanced relative to sinus rhythm. And you can see that also by the mean values. So sodium calcium exchange is upregulated in chronic AF and could contribute to DADs along with the ryanodine receptor hyperphosphorylation and leak. So you have hyperphosphorylation of the ryanodine receptor, which is super sensitive. It leaks in response to the increase in SR calcium that happens in diastole, particularly under conditions of adrenergic stimulation or rapid heart rates. And whatever amount leaks, the current that it generates will be amplified by an increase in the amount of sodium calcium exchange function. These were very interesting results. They were the first detailed study of calcium homeostasis in AF patients, but there's a limited relevance of these findings because it's widely believed, based on extensive evidence, that chronic AF is most likely maintained by complex reentry. And so the role of ectopic activity that happens in chronic AF patients is not clear, although you could imagine it would re-induce AF if it stopped. So then the question is, what happens in paroxysmal AF, where focal ectopy likely plays a bigger role? And Niels followed this up with a paper published 2 years later in Circulation, looking at samples from patients with paroxysmal AF. So these were patients with a history of transient AF who were not in AF at the time of their surgery when the samples were obtained. And again, you see calcium recordings, transmembrane potential here, not action potentials. And you see these spontaneous calcium leaks, which are very limited. They're really just oscillations in the control patient, and very important spontaneous calcium releases in the paroxysmal AF patient sample. So what are the mechanisms in these patients? So I didn't show you the SR calcium load measurement in chronic AF, but it wasn't changed. But here they found, interestingly, that the amount of calcium in the sarcoplasmic reticulum was increased in these paroxysmal AF patients. They measured that as the change in intracellular calcium on exposure to caffeine and voltage clamping. And they saw a significant increase both in the calcium amount and in the charge carried by the by the sodium calcium exchanger, which reflects the total SR calcium load. So the calcium load is increased in paroxysmal AF, which it's not in chronic. So why is the question? So that takes us back to our calcium cycling and to look at the uptake of calcium into the SR. So one possible explanation for accumulation of calcium in the SR could be reduced ryanodine receptor function. So it's not coming out of it very much, but we saw that the ryanodine receptor is leaky. So if anything, the SR calcium should be reduced. So the question is, why is it increased? So the answer presumably would be at the level of the calcium uptake mechanism, because there's a competition between SERCA and sodium calcium exchanger for the disposition of the calcium that's released in systole. If you have a hyperactive sodium calcium exchanger and an unchanged SERCA, that will tend to reduce diastolic calcium. If you have a hyperactive SERCA and no change in the exchanger, then that'll tend to increase SR diastolic calcium. So there are two possible mechanisms, two obvious possible mechanisms for increased SERCA activity. One is simply the straightforward, an increase in the amount of sarcoplasmic reticulum calcium ATPase. And the other, which was not changed. Okay, so the other, which I thought I had a slide here, but doesn't seem to show, so I think maybe it'll come up later, is, sorry. So I'm getting a little ahead of myself. So the first question they asked was, what is SERCA activity? How active is SERCA? Can we explain the increased calcium load by an increase in sarcoplasmic reticulum calcium ATPase activity? And to measure that, they used a kinetic approach. So they fired the cell regularly and then applied caffeine to get a large release of calcium from the SR. So what caffeine does is it essentially blocks in an open state the ryanodine receptor and produces a massive leak of SR calcium. And most of the SR calcium is leaked out into the cell, and you can use that to measure the amount of total cellular amount of calcium. The other thing that it does is, because the SR is now completely leaky, SERCA becomes non-functional, and the kinetics of decay show you what happens to the cell when SERCA isn't working. So the rate constant of the decay of the regular action potential mediated calcium transient reflects uptake by SERCA and exchange across the membrane by the sodium calcium exchanger. When you apply caffeine, the SERCA is no longer functional, and you're measuring the kinetics with SERCA blocked, so with the sodium calcium exchanger kinetics only. So theoretically, obviously, this should be slower because it results from only one process, whereas the action potential mediated calcium decay should be much faster. And they plotted the kinetics and measured the rate constant of SERCA activity by subtracting this from this. So this is subtracting this rate constant from this one. This one should be larger than this one. And this is NCX plus SERCA. This is NCX alone. And the difference should be the rate constant for SERCA. And they saw an increase in paroxysmal AF, suggesting that calcium uptake into the SR is enhanced. So what are the possible mechanisms of this function? One would be an increase in SERCA expression. The second relates to an inhibitory protein called phospholamban. Phospholamban interacts with SERCA, and in fact, when it gets phosphorylated, for example, by adrenergic activation, it dissociates from SERCA, and the inhibition is removed, and SERCA activity is increased. So, for example, in the fight or flight reaction, when adrenergic stimulation is enhanced, the hyperphosphorylation of phospholamban allows SERCA activity to be increased and to compensate better for the increased calcium that's coming in the cell through the increased L-type calcium current, and the whole system basically gets jazzed up. So what happens to these two phenomena? So the first point to note is, so they used Western blots to measure the amount of SERCA, which is this top set of bands, the total phospholamban, and the phosphorylated phospholamban at the serine and threonine sites. And you can see that the SERCA expression is not increased in AF. In fact, it's slightly reduced, but the phospholamban phosphorylation is increased. And that could conceivably explain the increase in SERCA function and SR calcium loading. So phospholamban hyperphosphorylation in paroxysmal AF could explain calcium loading by virtue of disinhibiting SERCA. So in paroxysmal AF patients, we looked then at the intra-SR calcium, what happens to the ryanodine receptor. So these are direct recordings of ryanodine receptor function in lipid bilayers. The ryanodine receptor is isolated biochemically and incorporated into lipid bilayers. This work was done in Zander Verenz's lab in Baylor. And here you see the closed and the open state of single ryanodine receptor channels. These are spontaneous openings. And you can see that in the sample from the paroxysmal AF patient, there are many more spontaneous openings than in the control patient. And overall, the ryanodine receptor open probability was substantially increased in AF, in paroxysmal AF. And then they looked at ryanodine receptor phosphorylation. I mentioned that in chronic AF, the ryanodine receptor is hyperphosphorylated. They, in fact, did not see any change. If anything, a slight decrease in ryanodine receptor phosphorylation. So the increase in the channel opening was not due to hyperphosphorylation in this case. The only change that they saw was an increase in the expression of the ryanodine receptor, and they have some theories as to why that might cause leaky ryanodine receptors. So ryanodine receptor function is abnormal with excess opening, and ryanodine receptor expression is increased, but ryanodine receptor itself is not hyperphosphorylated. Finally, they looked at the sodium-calcium exchanger, and they found no changes. So the conclusion of this work is the same phenotype can be produced by different molecular mechanisms. In chronic AF, the mechanism responsible for the spontaneous calcium releases and ectopic beats triggered by DADs is increased sodium-calcium exchanger expression and function, and ryanodine receptor dysfunction leakiness due to CAM kinase hyperphosphorylation. In paroxysmal AF, ryanodine receptor dysfunction happens, it's clear, but without hyperphosphorylation, and there's increased calcium load due to increased expression of SERCA. So actually, this is due to increased activity of SERCA, which is actually not due to change in expression of SERCA, but to hyperphosphorylation of phospholamban. So the key takeaway points from this section on atrial ectopic activity are that atrial ectopic activity is involved in initiating and possibly maintaining AF. Atria from AF patients show a tendency to generate ectopic beats through calcium-mediated activity. And finally, both chronic AF and paroxysmal AF patients manifest DADs and ectopic activity due to abnormal calcium handling, but with distinct mechanisms. I'm going to speak now briefly about AF perpetuation before we take a break. So I mentioned that ectopic activity landing on a vulnerable, acting as a trigger on a vulnerable substrate can produce reentry that perpetuates AF. And one of the important mechanisms involved in this is a phenomenon that was first described by Moritz Alessi's lab in 1995. So that would be 26 years ago, quite a while ago. It's known by the moniker AF begets AF, which very succinctly summarizes the mechanism in a way that can be understood. So what Moritz did was to take goats, put electrodes into the atrium, and then burst pace the goat's atria, something that anybody can do. They use goats because they're readily available and not terribly expensive and easy to keep in the Netherlands. So when you burst pace the atria, it goes into AF. Everybody knows that. But what they did, which was different, was they connected the pacemaker. The pacemaker also had a sensing circuit and could sense when the goat went into sinus rhythm. And when that happened, they immediately put them back into AF. So what they would then get is a very important combination. They could keep the goats in AF most of the time, while at the same time getting an ongoing log of how long they were in AF. And by the way, this is work that Philippe and Guido are doing in Bordeaux in a closely related sheep model. So what they found was after 24 hours, the AF tended to be more long lasting. And after 2 weeks, 85% of the goats were in sustained AF. So that shows us that just having AF itself changes the atria such that they can more easily maintain AF. So AF-induced remodeling, which is this component, and I'll come back to this later in more detail, is a major cause of perpetuation and progression of AF. So that if a person has an episode of, this is a diagrammatic representation of the natural history of AF from a typical non-real patient who has a bunch of temporary paroxysmal episodes, then has one persistent episode, and you see the persistent, and the AF substrate is shown diagrammatically by the vertical displacement. So the higher the point, the colored point is here, the greater the AF substrate is, and the more likely AF is to maintain. So the occurrence of AF itself changes the AF substrate such that it's more likely to maintain itself. Then when AF stops, in this case shown by a cardioversion, the substrate starts to wane until the next AF episode. And atrial rate relating remodeling is responsible, one important mechanism responsible for AF perpetuation. Now, in order to understand the determinants of reentry, and I suspect you've had this, or have you had this already in a discussion of electrophysiology? Yes, no?
So yes, they did.
Okay, all right, so I'll go through it very quickly. I'll go through it very quickly then. So this is an idea put forward. It's really a quantification of a much earlier idea put forward by Moritz Alessi in the 1970s. The idea is that the minimum path length that can support reentry is the distance the reentrant impulse can travel in one refractory period. And that's the product of time that it travels, which is the refractory period, minimal time, and conduction velocity, which is the rate at which it's traveling. So this defines the minimum path length for reentry, which if you induce functional reentry will establish how large the reentry circuit is. So in a normal atrium of normal size, the normal wavelength is such that AF is not spontaneously sustained. And if you induce AF in a normal person, it'll spontaneously self-terminate quite rapidly. But if you shorten the wavelength, for example, by decreasing action potential duration, which will reduce the refractory period, then you'll have more of these circuits and they're less likely to self-terminate and AF is sustained. So that's the classical view, and we'll revisit it later on. So shortened refractory periods will happen if the action potential is shortened, And that can happen from decreased inward current, such as decreased inward calcium current, or an increased outward potassium current. So a lot of research has been done by my lab and others to figure out how atrial fibrillation, or very rapid atrial activation, produces this perpetuation and remodeling. And I'm going to summarize that diagrammatically on this slide rather than showing you a ton of separate experiments. So the basic idea is that when someone goes from sinus rhythm to AF, they go from a rate of the order of 60 per minute to 400 to 600 per minute, which is a 10-fold rate increase approximately. So with each action potential, I already mentioned, calcium comes into the cell. If you have so many action potentials within a short period of time, you get cellular calcium loading, which is a threat to cell viability. So the cell acts to protect itself. And within minutes, it produces calcium-dependent calcium current in activation. So the L-type calcium channel is sensitive to the level of intracellular calcium, and if it starts to build up, the calcium channel starts to inactivate, the current decreases, and that feeds back negatively on cellular calcium loading. Over longer periods of time, you get molecular mechanisms that come into play, in particular, a downregulation of L-type calcium mRNA, but also possibly proteolysis and enhanced phosphatase action, that all of which affect the L-type calcium channel function, and you get a further reduction in calcium current. So when the calcium current is reduced, the amount of positive ions coming into the cell during the action potential plateau is reduced. There's fewer calcium ions coming in, so the cell repolarizes more quickly, the action potential shortened, and the cell moves from one to multiple circuit reentry, and that perpetuates AF. It makes AF more likely. So the key points regarding AF perpetuation and atrial tachycardia remodeling is that when AF is maintained, it causes electrical changes that promote its own maintenance. And a key component of this change is a reduction in action potential duration and refractory period, which result from the ion current adaptations to rapid atrial rates. Okay, so we're going to talk now about the next step in AF, which is progression. So we talked about how ectopic activity can initiate or even on its own maintain AF. We talked about how ectopic activity could act on a vulnerable substrate, which could be potentiated by atrial remodeling induced by AF or perpetuated. Now we're going to talk about the progression of AF, which is a fundamental process. So we know that many patients present with paroxysmal AF, which then becomes persistent. If it's not managed, tends to become long-term persistent or even permanent. Okay, so this diagram, which I presented to you before, which I'll go over in more detail now, represents a schematic notion of the natural history of AF. And so here we see these episodes of AF, which I mentioned, which produce an increasing AF substrate with time. And we'll come back to the form of this relationship. But in addition, the factors that promote AF worsening, increased likelihood, and increased resistance to therapy with age are shown here. Now, I didn't mention, I assume many of you know that age is the single most important predictor of atrial fibrillation. And the incidence is about below 1% until age 65, and by 80 or 85, the incidence goes to about 20%. So tremendous age dependence. And conceptually, we believe that the basis for that includes, of course, more time to have more episodes of AF, but importantly, the normal aging-related substrate, which increases with age, And if there's no heart disease, is in itself not so likely to cause AF. But on top of that, there's a remodeling substrate due to cardiovascular disease. And as we'll discuss later on, this cardiovascular disease that produces atrial remodeling doesn't necessarily have to be an overt disease like congestive heart failure or valvular heart disease that's really an important pathological entity requiring treatment. It could be something as apparently innocuous as hypertension, which causes left ventricular hypertrophy and changes over the time, or could even be cardiovascular risk factors, which, although in the short term at least, they don't cause overt cardiovascular disease, they still can act on the heart to promote the AF substrate. So I'm going to talk now about one of the early observations that pointed towards the kind of remodeling that can cause AF progression. And this study we actually did in around the time or just after Dr. Alessi's work, based on asking ourselves the question, it's fine to say AF begets AF, and that if you have AF, it'll promote its own substrate, but something has to cause the substrate for re-entry in the first place. What could that be? So we used one of the most important clinical causes of AF, which is congestive heart failure, and induced it in dogs. And we did that by putting a pacemaker lead in the ventricle and tachypacing the ventricle of dogs at 240 beats per minute for 2 weeks, and then reducing the rate to 220 beats for another 2 to 3 weeks. Oh, sorry, other way around, 3 weeks and then 2 weeks, total of 5 weeks. And then brought them to the electrophysiology lab and studied the atrial substrate. So typically in my lab, we study the atrial substrate by inducing AF, and in a normal atrium, AF usually self-terminates. If there's a substrate for AF, it usually lasts for varying, but often longer periods of time. So this slide contrasts the sustained AF substrate in an atrial tachycardia remodeled animal. So this is a dog that's been subjected to rapid atrial pacing with AV block and a pacemaker to prevent any rapid ventricular rate for 1 week, compared to a dog that's been put into congestive heart failure by rapid pacing the ventricles for 5 weeks. So you see these are atrial electrogram recordings. Both animals have quite irregular atrial electrical activity. And so clearly both of these interventions produce a substrate that mimics, that can promote or maintain AF. Rapid atrial pacing mimics atrial fibrillation. So this is a simple way, without having Alessi's sophisticated pacemakers, a simple way to induce atrial remodeling. And congestive heart failure, of course, mimics a cardiac substrate, a heart disease cause of the atrial fibrillation substrate. Now, if we look at the proper electrophysiology lab, so we induce and measure its average duration, control dogs was very short with rapid atrial pacing or congestive heart failure, much longer. So these are statistical significance versus control. And you can see that these aren't different from each other. So the AF substrate is of comparable intensity in both models. But when we look at the refractory period changes and the wavelength, there's actually no change in the refractory period in congestive heart failure, whereas in rapid atrial pacing, we see the expected shortening of atrial refractoriness and reduction in wavelength. So it's easy to understand why this model, the atrial fibrillation remodeling type of scenario, should promote AF by reducing the wavelength and allowing for more circuits. But why does heart failure produce a substrate for AF? Not obvious here. In addition, you see no change in global conduction velocity. So what how does heart failure cause AF? And we decided to look at the fine structure of atrial conduction and atrial histopathology, and we saw something that we found really quite remarkable. So these are Masson trichrome stains invented by a guy named Bernard Masson in Montreal, a histologist, very widely used. Cardiomyocytes are stained pink and connective tissue is stained blue. And you can see in control, very fine tissue septi separating the muscle bundles. And in atrial tachycardia, the AF remodeling type of scenario, same thing, no real increase in the amount of connective tissue. But here in the heart failure animals, you can see these large areas of atrial fibrosis separating muscle bundles. And the fine structure of conduction shows something quite interesting. So this is an isochrone map, which includes lines to indicate the regions that are activated within 10 millisecond intervals. So you see we're pacing here at the right atrial appendage. You see that the impulse travels quite quickly along Bachmann's bundle, no issues there. It looks very normal here. But on the left atrium, you see these regions of very, very slow conduction, crowded isochrones. This impulse is going in this area it's taking it about 30 or 40 milliseconds to go a distance that would take maybe 10 milliseconds in an unaffected region. With atrial tachycardia remodeling and the control condition, you see none of this. You see nice, smooth, symmetrical conduction. So we concluded from this work that heart failure causes fibrosis in the atrium, which impedes atrial conduction locally. The reason why the overall conduction velocity isn't changed is that there are zones which are fairly normal and allow normal conduction, but there are also local zones which have intense fibrosis and conduction slowing. So this work showed that fibrosis underlies the AF substrate in congestive heart failure, and clinical pathological examinations have shown that many patients with AF have fibrosis in their atria, particularly with heart failure, but also with valvular heart disease and other cardiac pathologies. And this is a clinically important paradigm because in most AF patients, the ERP and wavelength are not reduced. And so it's not so obvious why they have atrial fibrillation, which leads to a wrinkle on the initial Alessi theory of the wavelength, in that in these kinds of substrates, what we believe is happening is that although you have a normal wavelength, reentry gets stabilized by these regions of fibrosis. And you have a much smaller number of circuits, which are slower, and some fast irregular conduction that causes AF. In fact, if we go back, we can see that the activation rate in heart failure is much in the atria is much slower than it is in with the atrial fibrillation type of remodeling. So, and this is a reflection of the fact that you have larger circuits and a fewer number that maintain AF. And the reason they don't stop spontaneously is that they're stabilized by these regions of fibrosis. So we asked before how remodeling promotes reentry. We talked about shortened refractory periods, which could come from decreased inward current or increased outward current, but impaired conduction causing local conduction disturbances can do a similar kind of thing, but via a different mechanism, not necessarily by shortening the wavelength, but by causing local conduction blocks that stabilize reentry. And this can be caused by tissue fibrosis, impaired connection function, which we didn't study in this initial work, but is shown in a lot of AF substrates, and decreased inward sodium current. So heart failure then can lead to a fibrotic AF substrate. There are interventions which have been used to try to prevent the progression of fibrosis in heart failure, and there's some evidence that it works, but the evidence is limited. There's been an interesting application of this concept to obesity. So morbid obesity produces an AF substrate. Patients who are extremely heavy are much more likely to get AF. And Prash Sanders' group in Adelaide, Australia, it produced an animal model of obesity in sheep. So they overfed their sheep, and they looked at the number of AF episodes as a log of time, and both spontaneous and induced. So red is spontaneous, blue is induced, as a function of animal weight. And you can see that the heavier animals were much more likely to get AF. And they noted conduction disturbances in the obese sheep. So here are activation maps, much more sexy and sophisticated than the ones we had years before. But you can see again these prominent conduction disturbances in the overweight sheep and slowing in conduction velocity. And they looked at tissue fibrosis using a different kind of histological stain. We had used Masson trichrome. They used picrocereus red, and you can see in the obese sheep increased regions of red indicating increased fibrosis. And they saw an increase in the percent of fibrous tissue, both in the right and left atrium in their obese sheep. So obesity produces a clear mechanistic substrate for AF. And then they went on to study the effects of weight loss on the AF substrate in man. So this, I should mention, this intervention was not only weight loss, it was an intense risk factor intervention. So people reduced their alcohol intake, stopped smoke, or were encouraged strongly and followed up to lose weight, stop smoking, control their alcohol intake, control their blood pressure, and so on. But one of the major changes they focused on, which they were successful at achieving, was weight loss. So in the control group, the baseline weight in kilos, so it was 110 kilos. You can see it's significant. Oh, sorry, this is the waist circumference. 99 kilos at baseline in the intervention group and 101 kilos at baseline in the intervention group, a BMI of 32 versus 27. And you can see with weight loss, they went down to 80 kilos in the intervention group, but lost a little bit of weight, but not very much in the control group. And then when they looked at atrial fibrillation incidence, the number of episodes of AF substantially decreased in the weight loss group, the number of episodes and the total duration. So they've since done a lot of follow-up experiments and confirmed in clinical populations these results. And although not everyone has been able to confirm their results, there have been multiple studies that have, including a study of bariatric surgery patients who obviously lose a lot of weight. And if they have AF, their AF tends to get much better. So reversing obesity reverses the AF substrate, and controlling cardiovascular risk factors may prevent substrate development. So the key points in this section regarding progression of AF is that both heart disease and heart disease risk factors promote AF, mainly by causing atrial fibrosis. I haven't gone through the data for other cardiac risk factors, but there's now data on hypertension and sleep apnea that both of them seem to act by increasing fibrosis. Preventing or suppressing risk factors may help to prevent AF by preventing or possibly even reversing atrial fibrosis. So fibrosis, as I mentioned before, underlies the AF substrate in heart failure. It's an important paradigm. We just saw it also happens with cardiac risk factors. And a relevant question is whether AF itself can, if it's sustained long enough, whether it can cause fibrosis. So we know that patients with long-standing AF are much more likely to have atrial fibrosis and be very resistant to therapy. And a fundamental question that we asked, and others as well, is can AF itself, by virtue of the rapid rate or some other factor, cause or atrial fibrosis? So the question, in a sense, is can the AF induced remodeling be partially irreversible? So we know that the electrical substrate, like Alessi studied, in the shorter term, within a few days, is fully reversible. When you stop tachypacing, the AF substrate completely reverses. But whether, if you have longer periods of AF, that can produce changes that are irreversible, like atrial fibrosis, has to be asked. So Brett Burstein, who was a PhD student in my lab at the time, took rapidly paced atrial-derived cardiomyocytes and put them 24 hours in culture. So these were HL1 cells, which he put into culture and divided into three groups. One group was rapidly paced, group was non-paced, so there were cells there, but they weren't paced. And a final group, which he called group O, was a group that had the medium alone with no cells, but was tachy-paced to control for any possible effects of electrolysis or other electrode effects. And he collected conditioned medium from these three groups and subjected cultured atrial fibroblasts to this media collected from the rapidly paced cells and analyzed the fibroblast responses. So these are photomicrographs of fibroblasts in culture for 24 hours from group O, non-paced and 5 Hz paced cells. And you can see the cells look different. These are larger, they have prominent projections, and they indicate differentiated fibroblasts. So when fibroblasts get activated, they become a different phenotype, myofibroblasts, which are less proliferative, but produce much more collagen. And he measured alpha SMA, which is an indicator with Western blot. It's an indicator of myofibroblast differentiation. And he saw clear increase in the alpha SMA in the 5 Hz samples, as shown here. And also an increase in collagen production by the fibroblasts exposed to the rapidly paced cardiomyocyte medium. So rapidly firing atrial cardiomyocytes release substances that activate fibroblasts and cause them to produce more collagen. So you could imagine that if this goes on, if this is only a 24-hour limited in vitro experiment, but if this goes on in vivo, it could eventually produce fibrosis. And supporting this are the following observations. So this is from Moritz Alessi, now with mapping experiments on patients in the operating room. Moritz took a high-resolution electrode grid, applied it to the atria of patients in the operating room, obviously in sterile and completely controlled and consented way, and induced AF. So in a sinus rhythm patient who had no history of AF that was put acutely into AF, the activation patterns in the atria looked as shown here with, so this, you're looking at a small window of the atrium. Obviously, it's not the whole atrium. You can't expect to see all of the atrial activation, but you see broad waves of conduction without any evidence of substantial local block. In contrast, in the patient with long-standing persistent AF, you see that the local electrograms are much more, the local isochrone maps, I should say, based on electrograms are much more complicated. You see all these tiny crowded zones indicating regions of block, like we saw in the dog. And you see these corridors, narrow corridors of conduction and very complex patterns consistent with the development of fibrosis and separation of muscle bundles. So increased complexity of activation with many more reentry zones suggests longitudinal dissociation of conduction pathways as would be produced by fibrosis in patients with long-standing persistent AF. A colleague of Moritz's, Sander Verheulen, did similar experiments in the goat model, and you can see, again, similar kinds of isochrone activation maps with large isochrones indicating rapid conduction in a goat with only short-term AF, whereas in the goat with long-term AF, again, you see these very crowded isochrones and regions of very heterogeneous and slow conduction. So in the goat, you see with long-standing maintained AF, but no heart disease, which is important because in patients, you can't necessarily exclude that, an increased complexity of activation with many more reentry zones, just like in humans, and then looking at the fine structure of tissue with histology, and this is a different approach to looking at fibrosis. It's not a stain per se, it's a microscopic technique, but again, you see these bundles of fibrous tissue which are very prominent in the long-term AF goat. So as AF becomes long-standing, fibrosis develops, especially in the epicardium, and separates cardiomyocytes, creating conduction barriers that can stabilize multiple reentry zones. So the key points from this section are that long-standing AF itself may be able to cause atrial fibrosis, and it may therefore be important to control AF as early and effectively as possible to improve the effectiveness of therapy. And in fact, there's now a study which was published last year in the New England Journal of Medicine, which compared early aggressive intervention in AF patients as close to onset as possible with more standard therapy, and they found that early aggressive intervention and limiting the duration of AF episodes and the number of AF episodes improved the outcome in these patients. So I'm going to finish now with a brief discussion of the pulmonary veins. So as I mentioned towards the beginning, a seminal observation in the AF field by Michel Eyzaguirre was that the pulmonary veins were critical in maintaining AF. Especially, I should say, in paroxysmal AF, where pulmonary vein isolation is curative in most patients, but also play a very important role in longer standing AF. Now, when we talk about the pulmonary veins, we're not talking about the veins per se. It's really a misnomer. We're talking about the cardiomyocyte sleeve that surrounds the pulmonary vein. So if you look at the pulmonary vein region near the heart, you'll see that there's a cuff of cardiomyocyte tissue around each vein. And it's that cuff which generates the activity that is important in maintaining AF. So Sander Verheulen produced a very important study looking at the histopathology of the pulmonary vein cardiomyocyte sleeve. And he saw that the cardiomyocyte sleeve was not a solid cuff around the pulmonary veins, not a solid cuff of tissue, but involved regions of fibers that insinuated into the vein itself. So the blue tissue here is the vein mass itself. And the pink are cardiomyocytes. So you see these things that look almost like snakes. Bundles of small numbers of limited numbers of cardiomyocytes that can be separated from each other by pulmonary vein tissue. So they saw longitudinal fibers that were separated in places by the veins and isolated muscle bundles. So this structure turns out to be very important for the arrhythmogenic nature of pulmonary veins. And it's important both for ectopic activity and for reentry. And in fact, now we have lots of data that suggests that the pulmonary veins are not only important in AF maintained by sustained ectopy, as was thought based on Michel Eyzaguirre's very early experiments, but can also play a critical role in reentry and reentrant AF. So first we'll talk about ectopic activity. Now, this is a diagrammatic representation of the sinus node and the atrial muscle. Now you might ask, why am I showing you the sinus node? It's not really involved in atrial fibrillation. So the answer is that the sinus node is the prototypical cardiac pacemaker. And the sinus node has a very particular structure. It has poor electrical coupling with the atrium. The structure and type of connections from the sinus node to the atrium are quite different from that between atrial cells themselves. So this represents the atrial gap junctions, which have high density of connections, particularly 43 and 40, which allow for very rapid conduction. Node connection is indicated diagrammatically here by much poorer connection. And this turns out to be critical. It's critical because if you have a zone that's trying to create automatic activity at a rate greater than the surrounding tissue and drive the surrounding tissue, if it were tightly coupled to the surrounding tissue, then it would not be able to drive the tissue because the tissue would electrotonically interact with it and kind of almost voltage clamp it to the potential of the tissue. So if atria are sitting at minus 75 to minus 80 millivolts, and it's very important for the sinus node cells to be depolarized to about minus 55 millivolts, not far from their threshold potential, so they can have fairly rapid spontaneous activity. Well, if there was tight electrotonic coupling, the SA node would simply hyperpolarize to a more negative potential because of the electrical influence of the adjacent atrial cells, and it wouldn't work well as a pacemaker. So if we look at the structure of the pulmonary veins, you have these isolated muscle bundles embedded in pulmonary vein tissue, and they're relatively poorly connected to the left atrial tissue. They obviously are connected because they can drive it, but I believe that it's these weak connections that allow the pulmonary veins to act much more effectively as ectopic pacemakers. I mentioned that the pulmonary veins are also favored sites for reentry, and this also relates to the pulmonary vein structure. So this is work done by Melez Hosseini, who's one of the senior faculty at Lyric. She's actually the associate director of Lyric years ago, and she studied dog atrial preparations and did intensive mapping of the pulmonary vein region. And what she noted was that the muscle bundles of the pulmonary veins had much less organized direction than the left atrial tissue. So there would be longitudinal and then transverse muscles that had to be coupled to each other in order for conduction to happen. And she noted within the pulmonary vein sleeves zones of relatively slow conduction. So you can see these crowded isochrones where here is normal conduction. These are very small distances, 8, 10, 12 milliseconds. And here you see a crowd of isochrones where it takes 12 milliseconds to go a distance that would normally take 2 milliseconds. And then what happens with a premature stimulus is that that becomes even more exaggerated because now you're operating in the relative refractory period of the tissue, and you get regional block, very slow conduction around it, and you could easily imagine this resulting in local reentry. And in fact, investigators have now shown in humans the same kind of potential for extremely slow conduction and favored reentry induction in the pulmonary vein sleeves. So the abrupt changes and limited connections in pulmonary vein firing can produce functional conduction block and cause reentry. Finally, it's not only the structural properties of the pulmonary vein sleeves, but also their electrical properties at the ionic level that favor ectopic activity and reentry. And these are results from a study done in my lab now about 18 years ago, where we compared, this is by Joachim Ehrlich, comparing action potentials from left atrial cardiomyocytes with pulmonary vein sleeve cardiomyocytes. And you can see that they look quite different. This is the resting membrane potential in the left atrium. This is in the pulmonary vein sleeve. So the resting potential is relatively depolarized because of a smaller IK1 background current in pulmonary vein sleeves. The calcium current is smaller and the delayed rectifier repolarizing currents are larger. So the combination of smaller calcium current to maintain the plateau and larger outward repolarizing current produces a shorter action potential. So the smaller IK1 depolarizes the cell, moving it closer to threshold, so that if there is spontaneous depolarization, it'll reach threshold and fire more rapidly and promote ectopic activity. It also, by the way, a small IK1 favors calcium-related ectopy through mechanisms that I can explain after if you want, a little bit complicated. And the shorter refractory period, and there's also a smaller sodium, shorter refractory period, and there's also smaller sodium current because the sodium current is partially inactivated by the depolarized resting potential. So between the slowed conduction from the reduced sodium phase 0 current and the short refractory period, all of these electrical features, action potential features favor reentry. So the key points from these areas are that the pulmonary veins are a privileged site in AF pathophysiology, and that both in ectopic complex generation, but also in reentry, and that structural factors appear to play a central role, but ionic current properties may also contribute to the participation of